Good morning. I think we'll go ahead and get started. Thank you all for making the trek to this room so far away from everything else. Today we have an exciting session on airway stem cells, tools, targets, and therapies. I'm going to give you a very brief introduction on why these three things, tools, targets, and therapies. But first, here's the disclaimer. The CFF hasn't vetted anything I'm going to say, so I'm going to go rogue. All right, so path to a cure. How can we really get at this? In order to get at this, we need to take a multimodal approach, right? Not one person's going to be able to do this alone. And so we need to be able to develop tools to better understand the fundamental biology of stem cells. We need to develop tools to try to get genes and modify airway stem cells and develop induced pluripotent stem cells. And we have to identify the right cell types as well, so we can then move the needle a little closer to therapies, which in itself is a whole different ball game, as we heard about on the plenary on Thursday. So the speakers today are going to hit multiple facets of this, of tools, targets, and therapies. First, we'll have Dr. Amy Ryan from the University of Iowa talk about a lung on a chip and how we can learn fundamental aspects about airway stem cells and their differentiation from that. Then we'll have my favorite speaker talk about a new stem cell in the airway called the hillock and its implications. Then we'll have Dr. Paul McRae, also from the University of Iowa, talk about shuttle peptides for in vivo gene editing, and in this paper that was recently published this week. Congratulations, Paul. And then we have Dr. Finn Hawkins from Boston University talk about this very exciting approach using induced pluripotent stem cells, iPSCs, for transplantation and rescuing CF. And so the objectives of this session, one, is to identify novel tools for deciphering the dynamics of airway stem cells, regeneration, and restoration of physiology, specifically mucociliary transport. Two, to evaluate approaches for targeting gene correction to basal stem cells, both endogenous and induced. And three, to assess the advantages and challenges of both of these approaches, both endogenous and induced stem cells. The way we'll do this, the format of this session, is we'll have 20-minute talks, about 5 minutes for questions, and we'll try to bring people up and have a panel discussion at the end. But please ask questions in between at each session, after each talk as well. All right, so let's go ahead and get started first with Dr. Amy Ryan, who's Associate Professor of Anatomy and Cell Biology and Associate Director for the Center for Gene Therapy at the University of Iowa. So I'll hand it over to Amy.
Okay, thank you very much. I'm hoping I can follow that level of enthusiasm that this session got introduced with and keep that rolling throughout. So I'm going to talk to you today about tools. We're going to talk about the lung-on-chip technology that we've been working on in my lab. A little bit of a disclaimer, not a lot of this is very specific to CF, but I will give you context of how it will apply to CF research. So I have no disclosures. And I want to be upfront to start with. These are the people who actually did the work. So this was a collaboration with Anne van der Does's lab in the Netherlands and my now graduated postdoc, Jana Nauwert, who is a principal investigator at the Pioneer campus at the Helmholtz Munich now. She came to me from Emulate, which is the company whose chip technology that we're using in the lab, and wanted to apply it to ask relevant biological questions. So this is the chip. This is the Emulate Biosciences S1 chips that are commercially available chip systems that have two channels, basically an air interface channel that's in blue on the diagram and a blood interface channel that's in red. The advantages of the lung-on chips, or the chip models, is that we can get kind of tissue-level models here of the airways, that we can really investigate the environment. And we're going to eventually be using this to look at the concepts of cellular engraftment in different environments. So we basically, in these chips, we can put the epithelium, we can add endothelium, we can have an air interface on the apical side, we can have a basolateral blood interface that we can manipulate what we put through that blood interface. It's continuous microfluidic flows through the blood channel. We can do real-time imaging on these. We can analyze immune cell recruitment in real time, and we can analyze mucociliary function and clearance in real time. So there's a number of advantages here. This is the actual system. The left-hand side of the image shows you the equipment. The orb basically is controlling the gas and power and the stretch that we're able to add to the system. The Zoe is essentially the computer station where we can manipulate the parameters. And then you can see the chips are in these little modules that slide in and are controlled by that Zoe. On the right-hand side, you can see a little bit more detail of what the Airway chip looks like. So we've got the blue channel. We see that airway epithelium in that channel, so airway basal stem cells. It's also being done with alveolar type 2 cells. We see them in that channel, expose them to air and do a differentiation just like you would in a trans well at the air-liquid interface. We have medium that we can flow through the basolateral channel, which is the pink channel. Again, in there we can just put media. We can put media with immune cells in that. We can line that channel with endothelial cells and make it like a blood vessel. And then we can control the amount of airflow, the rate of airflow, and the amount of stretch we put in the system, mimicking different levels of the airway. So I like to see this as a little bit more translational in that we're having some functionality in this chip that more mimics airway function. So when I agreed to let my postdoc bring this technology into the lab and work on this, I thought this was a great system. And a couple of months down the line, we get our air-liquid interfaces up and running. It took 2 years to optimize the protocol to get the cells growing on these chips. And the biggest challenge is most of the chip technology out there is using the standard PET membranes with 0.4 micron pores, just like your trans wells. Epithelium differentiates great on those. These chips, to make them stretchable, are PDMS membranes, and they have 7 micron pores. Epithelium migrates straight through those into the basolateral channel, and you get no differentiation, you get no air-liquid interface. It took us a long time to figure out how to resolve that. This is now published, and I'm not going into the details, but as you can see on the image on the right, the no coating led to the migration of the cells. You can see them growing on both sides of that membrane. With our anti-adhesive coating protocol that we developed, we now have them staying on the apical surface and able to undergo air-liquid interface differentiation. So we then wanted to characterize what was going on in the chips. Are we reflecting the same level of differentiation, same cell types that we're seeing in our trans wells? And the answer is yes. We did immunostaining, we did cell counting, we looked at the surface area coverage, we did RNA, and really there was no substantial differences between the differentiation on the chips and the differentiation in the trans wells. We then wanted to look more into the dynamic properties of the chips. So we were looking at mucociliary clearance and ciliary beat frequencies. And again, the images on the left here are showing our analysis of ciliary beat frequency. We see that there was no difference in ciliary beat frequency. However, we did see after 14 days of differentiation, so this is an early time point for your air-liquid interface cultures, we saw that there was significantly more multiciliary genesis happening in the chips at day 14 than there was in the inserts, suggesting that maybe there's an acceleration of differentiation on the chips, which I will come back to later in the talk. On the right-hand side here, we then looked at the mucociliary clearance at day 14 of differentiation. And what we could see that correlating with the about 1/3 better coverage that we had, we saw an increase in directionality and speed and coordination between the cilia at this early time point. Day 14 in the trans wells, you really don't see a lot of coordination in the cilia beating and mucociliary clearance occurring at that time point, which you can see in the chips. We have directional flow already established. So we did a little bit more investigation into what was happening and to see if this really was a matter that the cells were differentiating and maturing a little faster in the chips. And you can see here we're looking at planar cell polarity developing. You can see that at 14 days in the chips, we're already establishing good levels of planar cell polarity, coordination between the cells, whereas in the insert, we're seeing no Van-Gogh staining on the cells, and it suggests that there's no coordination happening at this time point. So we feel that the dynamic flow that we're adding into the system is actually promoting a functional maturation of the multiciliated cells. So this is an analysis now of the airflow and stretch and how it polarizes the multiciliated cells. We looked at the expression of the different cell types in the controls, which is with no airflow and stretch, or with an airflow and stretch that was mimicking the level of stretch that you would get in the trachea. So we weren't going down to the small airways at this time point, but you could mimic the stretch that was experienced in any of the airway branching generations. And we did see some indication that there may be a change in the distribution of cells. We saw more club cells with airflow and stretch and maybe decreased basal cells, perhaps indicating that the differentiation is not mimicking the trachea as closely and maybe is mimicking a more smaller airway. But there was no considerable changes there. But what we did notice was that airflow and stretch really established directional flow, which goes along with the coordination and maturation of the epithelium. So on the left, you can see we're monitoring the trajectory of the beads, looking at flow direction. And you can see in the control there is some skewing towards the inlet, but no actual directional flow. But when you have airflow and stretch, the cilia actually beat very and align against the flow, which we thought was quite interesting. We expected that to maybe align with the flow, but it was very clearly going against the flow towards the inlet of the direction of the flow. And that's just the quantification of that difference in direction on the right-hand side. So then we wanted to dip into it a little bit more. Is anything changing about the epithelium? So we did bulk sequencing on the cultures here, looking again in the control or in the conditions where we had airflow and stretch in the chips. And some of the things I want to highlight is that we see quite a difference in the expression of extracellular matrix-related proteins, which suggests that there's a different niche being established in these chips. You can see the differences in here, but on the right-hand side, just highlighted a couple of major differences, collagen 1A1, fibronectin, and some matrix metalloproteases are quite considerably lower expressed in the chips, which suggests maybe changes in the environment and changes in the inflammatory environment that might be typical in different disease states as well. Oops. We also validated that expression at the protein level and saw that the secretion of these proteins in the supernatants that we're able to take were significantly decreased too. So it suggests that the cells are behaving a little bit differently in this different, more functional environment than they do in those static chips. And many of the genes that we see changed are associated with airway remodeling in a number of lung diseases, including asthma, COPD, and CF. So this we think is moving towards a more physiological tissue-level human models, and these chips really allow us to evaluate the functionality of the epithelium, the differentiating of our cells, and perhaps cell engraftment in a controlled environment where you can add different cell types into the mix. So we're now starting to move this chip into applying it to look at our disease states and see if we can create our disease on a chip. Now, we haven't got a lot of data on this yet, but this is just some of the preliminary data we have from using CF primary bronchial epithelial cells. I apologize for the differences in the colors on the images, but basically you've got ciliated cells in red on the top and in green on the bottom. You've got our non-CF chips on the top and our CF chips on the bottom. And although differentiation happens pretty well in both, what we did notice that when you add airflow and stretch and start looking at the coordination between the cilia, there seem to be some differences in the mucociliary flow patterns in the CF chips. As you can see on the right-hand side, you can see nice directional flow in the controls with the CF chips in the presence of the airflow and stretch. There's a little bit discoordination between the ciliated cells. We are not sure if this is a CF phenotype, but we have now done this on 3 different donors and seen very similar findings. And this is not something that we've ever seen or noted in the chips. You seem to get directional kind of circular flows in the chips, not in the chips, in the transwells. So we're interested in following up on this and seeing if that change in the maturation and the coordination between the cilia is a real phenotype. The other thing I want to kind of stress is why it's important to study multicellular systems. We've seen a lot of talks at this meeting who've looked at macrophages or neutrophils, but done it in isolation. And those cells are very responsive to the environment that they're getting those signals from. So we have started to establish co-culture methods, which we're now bringing into our chips, looking at macrophage interactions with the epithelium. And this is preliminary data that we've used cell lines. We've used the Kufi and the Newly cell lines just to control for patient variation there and have a CF line and a wild type line. And we have cultured these with wild-type macrophages and CF macrophages that we've isolated from donor lungs. These macrophages are isolated with a protocol established by my postdoc that we're able to do a full isolation and purification in 2 hours, trying to minimize any changes in those macrophages. This is just looking at secreted cytokines, and I've got 2 slides here that I really want to highlight why these co-culture are probably very important when we're thinking about how macrophages and epithelial cells function in disease states. On the left-hand side here, the red and green, and I apologize for anyone who's colorblind, I did not think of that. But the red and green on the left are the newlies, so the wild types, with on the lighter bars are the wild type macrophages and the darker bars are the CF macrophages. The same for the red, they are a CF epithelium with wild type and then CF macrophages. And on the right-hand side are our monocultures. So these are just the macrophages cultured by themselves. So one thing I want to highlight here is that if you're looking at the macrophages on the right-hand side, so the light gray and the dark gray bars, you can see that the first dark gray bar, which is non-stimulated, there is an increase in TNF-alpha from those macrophages in the CF. When you stimulate with them with the LPS, that increase is substantially higher and much more than what we see in our wild-type macrophages. However, then go back to the green and red bars, you can see when you have these cells in co-culture that the epithelium is actually suppressing some of that TNF-alpha release, suggesting that there's crosstalk and communication between those cells that is regulating that inflammatory environment that is being driven there perhaps by the CF macrophages. On the contrary, when we looked at interleukin-8, you can see on the right-hand side, while there is a signal coming from the wild type in the CF macrophages, when you do the co-cultures here, you can see that a signal is actually augmented significantly, and that when you have the epithelium, and especially the CF epithelium with the CF macrophages, you see that that is the highest release of IL-8 we're getting in our system. So for us, this suggests that when we're considering the inflammatory environment, the importance of having a multicellular system could be critical in really understanding what is going on. And this is something we believe the chip system can really help us kind of delve into. The beauty of the chip is you can do real-time sampling over long periods of time with very small volumes. So we can analyze a lot of these phenotypes in real time. This is just another preliminary experiment that I want to highlight. We all know that the CF airway is highly neutrophilic. That is highlighted in these images from tissues taken from CF lungs. On the right-hand side, you can see our chip experiments where we're looking at neutrophil recruitment. These are just epithelial cells. There is no endothelium in the basolateral channel. We're putting freshly isolated neutrophils from peripheral blood in the basolateral channel, labeled with a red. And we can see that after 30 minutes in the CF, which is the lower 3 panels, you can see neutrophils are starting to migrate up to and through the epithelium. And by 90 minutes, we're seeing them appearing on the apical surface. In the wild types, we don't see any or very little recruitment of the neutrophils. And just to show at the bottom there, those are the neutrophils in the wild type that are still remaining in the basolateral channel. So again, this suggests that the epithelium is regulating that neutrophil recruitment. And there's something specific to the CF epithelial cells that is retained from basal cells in culture that is influencing neutrophil activity. So that's all I'm going to present on the chip. In the last few minutes, I want to kind of move into it's a publication we have under review right now, but it's looking at functionality of the airway epithelium and how do we get that against in vivo function? You know, how close are we actually modeling the airways in our dish? So we started looking at mucociliary clearance and the cell types, and most of the data that's published is actually doing from histology or from transsections across the epithelium. We've not actually quantified the area of the luminal surface that is covered by each cell type. So this was kind of a summary of the literature that's out there, and typically it's showing a change in the distribution of ciliated cells, especially as you go proximal to distal down the airways. So we took human lungs and rat lungs and dissected out branching generations 1 through 6 in the humans and 1 through 5 in the rats, and then basically did live staining on these and live imaging of ciliary clearance and calculated a lot of different parameters. So first we looked at this, the luminal coverage of cell types, looking at ciliated club and goblet cells. We then quantified that down the branching generations, and that's summarized in the graph in the middle. We can see that human ciliation is actually pretty consistent and around 90% through the top 6 branching generations. That is considerably different to the rat and the mouse airways, which increase in ciliation as you go down the proximal distal axis. We looked at the secondary cell types and then broke those out into mucus, MUC5AC, and CTB1A1 expressing club cells, and quantified the double expressing and the single expressing on the right-hand side. What we did note was that there was a considerable amount of cells that were both co-expressing MUC5AC and CTB1A1. We then looked at cilia function. So we did cilia beat frequencies and clearance speed, again, at each branching generation in these live tissues. The one thing that we did notice is that the clearance per beat, although there was no significant difference in beat frequency and clearance speed in the humans and the rats, the amount of clearance you got per beat of the cilia was actually substantially different between human and rats. We then wanted to take all of these measures that we were getting and put them into a model where we would be able to take some parameters and based on that predict from our in vitro models how these would apply to the in vivo setting. The computational side of this is way beyond me. This was my postdoc's work, talented computer scientist. But we basically took cell level input metrics. We took the cilia length, cilia beat amplitude. We looked at the power stroke and the recovery stroke. We took the tissue level metrics of looking at ciliated cell coverage, the beat order, which is the coordination between the cilia, and then we did some measures of the distribution of the ciliated cells, looking at patchiness and crystalline order. So that's really how they're organized, whether the ciliated cells are clustered and how the secretory cells are interspersed with the ciliated cells. And we then created tissue level output metrics, including clearance per beat and clearance directionality. On the left is just a summary of some of those measurements, again, looking at the human and the rat. And on the right is our predicted model, with the lighter area being the error of the model predicted. And what you can see is that our human benchmark is highlighted by the red circle, sits very nicely on our model line, and the rat benchmark also sits very nicely on the rat model line with the green circle there. So this will predict, based on ciliated cell coverage, what the function and clearance per beat could be. We then wanted to test whether this would fit with our in vitro models. So we took standard transwell air liquid interface cultures, cultured these in the many different medias that are available, which are listed along the top. And again, did the same parameters, looked at this airway surface coverage of the different cell types. Then we looked at all the parameters of ciliated cell function. And what you can see here is there's a lot of these medias that we culture our human airway epithelial cells in and differentiate them actually benchmark much more closely to the rat airways, with pneumocult is the one there that is actually hitting the human benchmark for the trachea. And again, we plotted these on our model, and you can see how well the different systems align with what we see in our in vivo or ex vivo tissues. We then went into published literature and pulled out more information. We took more information from other studies we've done and put all of those different models into the system. So here we have our chip data at day 14, which is highlighted by that blue circle. And you can see that's aligning somewhere between the rat and the human. But we're able to benchmark each of our different systems here against what we expect. We also included in here an IL-13 treated airway, which should have significantly more goblet cells, less ciliated cells, and you can see that. Are you seeing, can you see my, there we go, yeah. You can see that up here where you can see that it's a very small dot indicating there's much more goblet cells than club cells here, and very few ciliated cells through its distance on that y-axis. So we believe that this is a system where we can actually do a little bit of translation from our in vitro models into the in vivo setting, and we're going to be applying this to look at maybe changes in ciliogenesis, cilia-related genes, or diseases where mucociliary patterning is changed, and try and benchmark how well we're modeling that in the dish, and then thinking about gene correction. How many cells do we need to correct to get sufficient mucociliary clearance that would map well towards what we would like in our in vivo setting? So just to wrap up and summarize, I hope I've shown you that chips can provide an alternative human in vitro model that really allows for directional airflow. We can integrate multiple cell types and look at tissue level cellular interactions that are actually fairly easy to dissociate and analyze separately in this system. The airflow and stretch do stimulate maturation of the epithelial differentiation, and that we get a functional mucociliary clearance that orients against the airflow. And I hope that showing at tissue level models can highlight differences potentially in disease states such as CF that we may be not able to see in other systems, and that the immune environment that is changed as a result of disease is something that we can model well in these tissue on chips. So with that, I just want to acknowledge my team, my funding, and a lot of this work was done in collaboration with the Cystic Fibrosis Stem Cell Consortium, who are all listed on the right-hand side, who have been great people to bounce ideas over the last 7 years I've been working with them. So thank you very much.
Ooh.
Are there any questions?
Actually, I'm going to ask a real quick question first because I can. Such interesting works. What a cool system to ask really fundamental questions. The one question I had was on the establishment of the directionality on the chip. You mentioned both airflow and stretch. If you take away one, if it's just stretch or if it's just airflow, which one sets up that PCP? I think that's like a fundamental developmental biology question that's super interesting.
We haven't done that yet.
Oh, cool. Okay, look forward to it.
Yep, we're seeking more funding. The downside of chips is they're not cheap.
Gotcha.
So we had to refine what we did on them. So we haven't separated them out, but I think that would be interesting. We'd also like to reverse the flow and see if the directionality changes if you reverse the flow and if the cells respond to that.
Fascinating. Great.
Thanks very much. Really interesting talk, Stephen Rose, CF Foundation. I was curious about the cilia setting up their motion against the airflow. That wasn't initially what I expected. I think you said you were surprised too.
Yeah.
And we had often conceived that if we could set up liquid flow across a culture, that might actually train the cilia to all coordinate and line up. Have you tried liquid versus airflow? And what do you speculate is the driving force behind this opposite directionality that you observed?
Yeah, we haven't tried that yet either. And speculation is about all I can do right now. I actually really don't know. And that's something we want to work out in the system, is what is the key signal that is driving that orientation and the alignment of the cilia. So yeah, a question that we're trying to answer, but a good question.
Hi, Amy. Nice to talk. Kara Fauché, CF Foundation. Have you ever looked at turnover in the fully differentiated epithelium in the chip? And is there any measurable turnover?
Yep, we haven't looked at that yet either. We've only taken these chips out 3 weeks and done the analysis up to 3 weeks, so we haven't looked at airway turnover at all.
All right, thanks.
Thank you. Comprehensive talk, and sorry to bombard you with ideas here, but you mentioned you saw a trend towards a difference in the mucociliary transport between CF and healthy cultures, right? Have you also tried applying ETI and seeing if it can restore the order?
Yeah, the answer is no, we haven't yet. We would have to then make that a static chip just because the amount of inhibitor that would be flowing through the system because it's continual flow, but we haven't done that, but that would be great. I'd say the CF work that we're doing on the chips, we've really just begun. That dyskinesis between the cilia there, I'm not sure what is driving that or whether it is yet a real observation. We need to really thoroughly validate it because it's not something we've observed in trans wells in the past. So we want to make sure we're looking at a real phenomena there, but looking at ETI would be a great advance on that, yeah.
Thank you.
Tom? No?
Go ahead. Yeah.
Okay, mine will be very short because I think you might have already answered it. For the emulate system, are you able to model bidirectional airflow? And will that impact the cell composition and the ciliary beating parameters that you're seeing?
Yeah, so that's what I wish we could easily do. You cannot reverse the flow, but what we could do is reverse the chips, I think. We're trying to figure that out right now. But the airflow is just unidirectional at the moment. We can't pump it back the other direction. But I'm sure there's a way we can do it. We're working on that, yeah.
Great talk, Amy. So, is this on? You showed some rat data, and I'm curious about if mice also work. And the question behind the question is if you might consider those genes that regulate left-right axis orientation, because I think, at the risk of looking stupid, but I think they are important in the direction in which cilia beat and that might actually change airflow. I don't know if you thought of that or.
Not yet, but sort of going back to your rat, we did it in rat and human. There is already a paper out there that has done a fairly similar analysis in mice from the Prakash lab. It's published in Nature Physics. They did some of these parameters in mice. The mice look very similar to the rats, different to the humans. But yeah, we haven't really started dipping into it yet. There's a lot of mechanistics we really need to kind of elucidate in the system.
The nice thing with using mice is you have access to all those mutants, and you could look
Yeah.
at those
Yeah.
fundamental questions.
Hi, Amy, here. Roland Kolbeck, Spirovent. Great talk. Did you look at tight junction formation on that chip model and how it may compare to the static cultures and how that may relate to, again, viral vector transduction of airway epithelial cells?
Yeah, so we've not looked extensively. We've done occludin staining. It doesn't look much different to the chips at this time point. We haven't really gone beyond kind of your standard stains and looked more detail at which tight junctions are expressed. We have looked at a couple of claudins. Claudin 18.1 actually it seems to be expressed in the human airways. It's not expressed in the mouse airways. It's in the mouse alveolus. So we're seeing some differences between the human and mouse, but not necessarily between the chip and the transwell. But again, we haven't gone into a very detailed analysis of that, and there may well be differences. Yeah.
Maybe our last question.
Yeah, very nice talk. I'm Javier Campos from UAB. And my question is, since the airflow direction is both ways in the real life, do you think, but the airs are different. One is charged with oxygen, the other is charged with CO2. Maybe you can test air with oxygen or air with CO2, and then maybe the system is sensing the concentration of oxygen because the flow of itself is bidirectional.
Yeah, so in the system, we can modulate what is in the air composition, but currently we can't change the directionality with using the system as it is. I'm sure if I can find some talented engineer, they may be able to adapt that system so we could look at bidirectional flow and maybe have that coming with different airflow. So, I mean, that's a great suggestion. Right now, the limitations of the current system don't allow us to do that. Yeah.
Thank you.
Excellent. Well, thanks, Amy. Such interesting work.
So I have the pleasure of introducing our next speaker, Viral Shah. He's an assistant in medicine at MGH, and he is going to talk to us about the airway hillock cells.
Awesome. Start this timer. Great. Thanks, Amy. Thanks for the program committee for allowing us to have this session. I would thank the organizers, but that'd be not appropriate here. My only disclosure is I have funding from the CFF through Leroy Matthews Award. So today, I'm going to just lay out what I'm going to talk about. I'm going to give a brief background. I'll talk about hillocks, what this is in the mouse airway. Then I'll talk about what it is in the human airway, what we know so far, the next steps, and then I'll propose a model to kind of summarize what we've learned so far. So this is how I learned what the airway epithelia was and what we thought for the longest time, that you have a pseudo-stratified epithelium, meaning all the cells touch the basement membrane, even though it doesn't look like it. And it's primarily composed of these common cells, secretory cells, basal cells, and ciliated cells. And then this paper that came out of the Rajpal lab and in another lab really changed the way how I think about the airway epithelium. That there's these flavors of common cells, and that these rare cells exist, specifically the CFTR-rich ionocyte. And that's very exciting. There's been some great talks this conference on what the cell is and what it does. But they also found this cell in this paper called the hillock. And so I'm not going to talk about the ionocyte today. We're going to focus on the hillock. And to be honest with you, the Rajpal lab really didn't know what to make of the hillock when this was discovered. And the ionocyte really took the limelight. It's literally in the title of the paper. So today I'm going to share with you what we've learned on the hillock. So hillocks are not just cells. Hillocks are unique structures. And so first, let me orient you to what we're seeing here in this airway. So first, we're going to talk about mice. So we took a trachea, excised a trachea from the mouse, cut along the anterior aspect, anterior membrane, and then unroll the trachea and lay it flat. And that's what you see here. The anterior sides are cut on both sides laterally, and you have the posterior membrane and cartilage rings. And what we find is that hillocks have characteristic locations along the posterior membrane here and over cartilage rings. And we're identifying hillocks in this explant based on the expression of keratin 13. And in mice, keratin 13 is uniquely expressed in hillocks. And we'll talk about humans a little bit later. Hillocks are also stratified squamous structures. And so let's take a cross-sectional image of a hillock. Here we have ciliated regions, denoted here by a ciliated tubulin in magenta, atub, and that's surrounding the hillock. But the hillock itself doesn't have any ciliated cells, it's aciliate. And you can see that there's two layers of cells here. A luminal layer that's enriched in keratin 13, and a basal layer that also has keratin 13, a second layer below it. And that's pretty evident by looking at the DAPI staining here, the nuclear stain. But things can look stratified, but not be, hence the name pseudostratified. So we wanted to be really rigorous with this, to really determine if this was stratified. So to get at this, what we did is we expressed a photoactivatable GFP across the entire airway epithelium. And so the GFP at first wasn't activated. What we can then do is stimulate with a certain wavelength of light to activate the GFP in a single cell. And that GFP would then fill the entire cell, and we can get morphology. And that'll help us understand whether hillocks are truly stratified or are they actually pseudostratified. And so that's what we did here. You see photoactivatable GFP in green, and you see the basement membrane measured by collagen autofluorescence by SHG here in gray. And this is a secretory cell. You can see the secretory cell actually kind of looks like the cartoon, so we're doing a good job there. And it's pseudostratified. It's touching the basement membrane. Then we look at a hillock. Again, photoactivatable GFP in green here, the basement membrane in gray, and it looks like it's floating. But the hillock isn't floating. There's actually multiple cell layers here. But the luminal layer, this top layer, is not touching the basement membrane. So the hillock is truly stratified. It's a stratified structure in the normal airway. Now, this is kind of weird. The airway is not supposed to be like this. It's pseudostratified. But we have other epithelium in our body that is stratified. And the skin is a great example of that. It's a stratified structure. So we wondered if the hillock would have similar properties to the skin. The skin does an important role. It protects us from the extracellular environment, or the external environment, sorry. And so we wondered if hillocks would have increased barrier and be able to resist injury. Now, one thing I teach all the pulmonary fellows is that everybody aspirates. So one of the common injuries that we see is aspiration of gastric acid. And so we wondered if hillocks would survive this. So we took a mouse trachea, dunked it in 80 millimolar HCl, and then stained for dead cells in magenta here, and K13 to identify the hillock. And you can see the hillock here at the center, identified by green K13 staining, and dead cells all around the hillock, all the pseudostratified epithelia, but very few cells in the hillock are actually dead. We can quantify this here on the y-axis, dead nuclei per millimeter squared, pseudostratified, surrounding the hillock, compared to the hillock resists acid. Now, we're exposed to other environmental pathogens, for instance, other injuries. One in particular is viral infections. And so we wondered, would the hillock be resistant to flu? So here we see staining for flu nuclear protein. It's abundant here in the pseudostratified epithelium, but very few cells in the hillock get infected with flu. And we can quantify this on the y-axis, pseudostratified hillock is protected. Then we wondered, what other injuries could the hillock survive? One thing that we do clinically is actually freeze part of the airway to do a biopsy, cryobiopsies. And so we wondered, would the hillock survive this? So we took the mouse trachea, put it in DMEM, stuck it in the -80 twice, and then stained for dead cells. And that's what you see here. And you see a number of dead cells around the hillock, but the hillock itself, very few cells are dead. Quantified here on the y-axis, dead cells per millimeter squared, pseudostratified. The hillock resists freezing injury, cryo injury. So what happens after the hillock survives these tough conditions? Well, it then goes on to resurface the airway epithelium that had died. And so here we see hillock in green. This is actually a fourth injury, naphthalene toxin. It's a smoke toxin analog, and at this doses kills a lot of the pseudostratified epithelium. And what we see here is that over time, over days, the hillock is expanding after the pseudostratified epithelium has died to resurface the airway. And we wondered what's going on here. Are the basal cells replicating? And so to get at that, what we used a P63 Cree-ER mouse. So this mouse is a conditional mouse that allows us to label the basal cells. And the reporter here we used was a mosaic reporter, similar to the Brainbow mouse, except this one, all the basal cells would be individual colors. And so this allowed us to be able to track individual basal cells and their clones and how they expanded. So here we have, just to remind you, what we see where normal hillocks are located, posterior membrane and cartilage rings. And then after injury, we can then track what happens to the basal cells. So 3 weeks after severe denuding injury, what we see are these cells that are streaming out of the hillock. You see all these different colors. Let me zoom in on one here. And remember, each color here is a different stem cell clone. So all of these blue cells came from a single basal stem cell clone from the hillock. And see, what we see is thousands of cells. Actually, if we look at cell count over clone size, sometimes we see tens of thousands of cells of a single clone streaming out of the hillock. And so the hillock undergoes massive clonal expansion to regenerate the airway epithelial surface. Now, it expands over the epithelium, but does it just stay there as a squamous protective layer? It actually ends up differentiating into all 6 different epithelial cell types. So here we have the clone label in green, and I'm staining for all the different epithelial cell markers in magenta. So we have keratin 5 for basal cells, aciliated tubulin for ciliated cells, CC10 for secretory cells, GNAT3 for tough cells, which is important in mouse, PGP9, neuroendocrine cells, and BSND for the ionocyte. And so the hillock, after resurfacing the airway epithelium, differentiates into all 6 different epithelial cell types here. So let me just take a second to pause and tell you what we've learned about the mouse. And so what we're able to do here is define the hillock. We can define the hillock based on its location, here along the posterior membrane and over cartilage rings, its structures being a stratified squamous epithelium that highly expresses keratin 13 here, and its function. It resists injury and then regenerates the airway epithelial surface, differentiating all the epithelial cell types. Now, the million-dollar question here is this just a mouse thing, or do hillocks also exist in humans? So to get at this, we took a human large airway, explanted it, and stained for keratin 13, and that's what you see here in brown. And over cartilage rings, we see large patches of keratin 13, very reminiscent to what we saw here in the mouse. We can then do cross-sectional imaging of this. This is the mouse, to refresh your memory. And here what we see in the human. Again, we see keratin 13 rich region, that's aciliate, here are ciliated cells, and that they look squamous. This is kind of small, so let me blow it up for you. So here we have H&E, and a few sections later, we have the immunofluorescence. Again, keratin 13 rich region here, and we have a gradient, a transitional zone that ultimately goes to our ciliated tubulin here in magenta. When we look at the H&E, it's a little bit more evident here. You can see these cells that are very squamous looking. They're flat, very reminiscent to what we saw in the mouse. And so we see these regions in the human, but an important functional property of the hillock is its injury resistance. So do these regions also survive injury? Now, in order to be able to get at this, we had to be able to identify hillocks live, right? That was all fixed tissue. So to get at that, what we did is we used WGA to stain cilia. It binds some of the sugars on the cilia, and then we can identify the regions absent cilia. And those are putative hillocks. We can then take regions right next to them that have cilia, and then isolate the basal cells from the putative hillock regions and from the ciliated regions, and culture them. Now, what I'm not showing you here, but we went into a great depth to try to understand what are the differences between the basal cells in the hillock and the ciliated regions. And it turns out there's different sensitivities to retinoic acid metabolism. And so what we found is that we can bring out the hillockness by using retinoic acid inhibition. So we then cultured these to ALI in the setting of retinoic acid inhibition. And the basal cells from the hillock differentiated and became hillock-like, while those from the ciliated cell region became cilia, or your normal pseudostratified epithelium. And that's what you see here with an en-face image. Keratin 13 in magenta and a ciliated tubulin in green. And the hillock basal cells make hillocks in vitro, while the basal cells from the ciliated region make all the different epithelial cell types, including ciliated cells. Those hillocks, in vitro hillocks, actually have signs of increased barrier function. Here, if we look at transepithelial resistance on the y-axis, hillocks compared to the control, they have a tighter barrier. So then we asked, does it also survive injury? So we looked at flu, we looked at cryo injury, and we looked at acid injury. And in all three conditions, hillocks survive, while the pseudostratified in vitro cultures don't. So let me walk you through that. So here we see keratin 13 and flu and P nuclear protein in green. And we see here, compared to control, hillocks don't get infected. Measuring the number of cells infected per millimeter squared, control hillock, human hillocks in vitro resist flu. Cryo injury, very similar here. Nuclei are in green, dead cells are magenta. You can see maybe a dead cell here. Compared to control, many more dead cells. Quantifying it here on the y-axis, dead cells, control, hillocks resist, cryo injury in vitro. Acid, same thing here. Nuclei in green, some magenta dead cells in the hillock, less compared to what we see in the control. When we quantify, human hillocks in vitro resist acid injury as well. Now, humans are always more complicated than mice, and one of the challenges here is that not everything we see that's keratin 13 positive in the human is a hillock. And so here you see, this is a ciliated tubulin in magenta, keratin 13 in green, and nuclear stain in blue. And what you see here is that this is a ciliated region. It's a pseudostratified ciliated region. And you see that there's these blazing keratin 13 positive suprabasal cells. And this has been found in single-cell RNA-seq and characterized that way. But the question is, are these related to the hillock? Are they not? What's going on here? And clearly, we have more steps to try to understand what exactly this is. But the important part here about the hillock is that you need three things to be able to determine what it is. You need its structure, you need the gene expression, and you need the function. And I think to get at this in the human, we need to identify and interrogate all three of those things. And so the approach we're taking in the human is to use spatial sequencing plus functional assays on relatively similar samples from the same region to better characterize what are these K13 suprabasal cells, what's squamous metaplasia, and what exactly is a hillock. So that work's ongoing. Also, I've only shown you here the large airway, both in the mouse and in the human. So another question here is, do hillocks exist throughout the airway tree? And so that's ongoing work as well. And the last and interesting question here is, can we get vectors into the hillock? And so this is preliminary data as well. Here we have a mouse explant stained with a ciliated tubulin, and then keratin 13. And you can see kind of the overlap. The aciliate regions in here have keratin 13 over the classic hillock posterior membrane and cartilage rings. And then when we try to get AAV GFP into it, you can see that there's also these regions that are completely absent, AAV GFP. And if I overlay the hillock, it seems that the hillock is also resistant to viral transduction here as well. And so this is the first aim of my K08, trying to understand this mechanism so we can get at this. All right, so there's still a lot of work to do, clearly, but I think we've learned something important here about the airway. I just wanted to give credit that this is built on a lot of strong foundation in the literature. Specifically, previous work that showed there's a heterogeneous population of basal stem cells, some that are unipotent, primed to differentiate into squamous, and that are located on the dorsal epithelium and over cartilage rings in aciliate regions. And so I think what we ended up doing is kind of putting the puzzle pieces together and then finding out that this elephant is the hillock. So let me just end by proposing this model for you and summarizing what we know. So the hillock is a stratified structure that expresses keratin 13 that It resists denuding severe injury while all the other airway epithelial cells die. The hillock then goes on to expand, regenerate and resurface the epithelium, and differentiate into all the different airway epithelial cell types. Great. Lastly, I just want to thank all the people involved. This was truly a team effort to get this done, specifically my mentor, Jay Rajgopal, the other co-first authors on this manuscript, Brian Lynn, Chaim Chernoff, and everyone else in the lab. Particularly, I also want to thank our funding, the CFF, the NIH, LungMap, and also New England Donor Services for the gracious donation of a lot of these tissues that weren't suitable for transplant. Great, and I'd love to answer some questions. Please, go for it, Esther.
Hi, Esther Vladar from the University of Colorado. Awesome, really, really enjoyed it. So thinking about your AAV experiment, is what you're maybe considering to switch to an AAV serotype that targets skin?
Yeah, that's a brilliant idea. We tried a number of different AAV serotypes, and this one worked the best in the pseudostratified epithelium, but maybe there's a better one that works in the skin that could be used here. That's a brilliant idea. We could also think about directly evolving the capsid as well, which is kind of in the works too. Yeah.
And just as a quick follow-up to that, like the desire to have an ALI system that can make hillocks for you, were you ever able to get hillock ALIs out of sort of the non-hillock ALIs by modulating retinoic acid signaling or something along those lines?
Yeah, brilliant question. And so the part I didn't really get to go into here was that the hillock basal stem cells have a higher sensitivity for retinoic acid. They're just very different enzymes in the pseudostratified basal cells compared to the hillock basal cells. Turns out pseudostratified epithelia want to make retinoic acid, while hillock basal cells want to break it down. And so what we found was that they have a higher sensitivity to retinoic acid inhibition. But if you crank it up, if you add a lot of retinoic acid inhibition, you could even turn the pseudostratified epithelium hillock-like. So you could potentially do that. I would probably do an extended dose response curve to get at that, though. Yeah.
Very nice, thanks.
Of course.
Ian Thornell, University of Iowa.
Hey, Ian.
Hey, how's it going? So I'm interested in your photo-activated fluorophore experiment.
Yeah.
Did you activate that by single photon or multi-photon microscopy?
Yeah, great question. We did it by 2-photon microscopy. I used a wavelength of about 800.
So did you activate in kind of like succession through the layer to see how many cells were stacked, or have you done that?
Yeah.
How high is it?
No, that's a cool experiment. We have some preliminary data to see how many layers there are. It seems to vary depending on the structure of the hillock. There's at least 2 layers. There might be some times where there's more than that. That's a really interesting question. Yeah.
All right, thanks.
Yeah, thank you.
Hi, Wenjie Ryu from University of Iowa.
Hey, Wenjie.
Well, great talk. I have a question about the distribution of hillocks in the airway tree. Is that enriched in the trachea or in the bronchus or lower airways? Another question is how evolution maintain hillocks in certain position of the airways. Do you know that?
Yeah, love those questions. So right now we've only looked in the large airways for the hillocks. We're in the process of going deeper down to the smaller airways as well, both in human and mouse less so since it's less relevant for us here. Why these locations? Why the posterior membrane and cartilage ring? That is such a fundamental question. We don't know the answer yet. The esophagus is right posterior. I don't know if that has something to do with it, but it's also a squamous epithelium. So it could be interesting to think about. I think there's a lot of developmental biology we could learn from that. Yeah, great question.
Hi, Margarida Amaral from the University of Lisbon, Portugal. Hi. Congratulations, this was great work.
Thanks.
My question is, for the human tissue, do you have any other biomarker than keratin 13? Because it seems to be non-specific.
Yeah, great question. So we're still working on doing that with spatial and the functional assays, because I don't want to tell you a marker that I don't know satisfies everything for a hillock. I think Esther Vladar, who was just here, had some great work on looking at in the nose and might have some markers as well. But that's in the works. I'm happy to share when we figure it out.
Thank you.
Of course.
Ashley Cooney, University of Iowa.
Hey, Ashley.
Very nice talk.
Cool, thanks.
At the beginning, you showed KRT4 as another marker for HILOX. Have you looked at KRT4 with KRT13, and are those expression levels changing through the way that the cells divide and different stages of regeneration?
Yeah, great question. So I think we're looking at, initially we'd seen keratin 6 in addition to keratin 13 for the HILOC. In terms of development, we're in the process of looking from postnatal day 1 onwards to how this develops, which turn on first, and so on. One of the things that I'll caution everyone about, specifically with keratin 13 and keratin 6A, is that they're injury responsive genes. So you put them in alley and they turn on. And so they're not going to be your best markers if you differentiate the cells or damage them at all. And so what's important about this is that this is happening at baseline, at homeostasis. So we still don't have great markers. There might be some of the serpins, for instance, some of the desmosomes, another instance, that could help identify HILOC-specific cells in damaged regions, but damage is going to confound some of this. So that's an important point I just wanted to mention.
Thank you.
Of course.
Hi, Hillary Valley from the CFF. You showed some really compelling data about HILOX being resistant to various types of injury. And I was wondering if you could comment more on the mechanism of that. I mean, HILOX are stratified, as you showed. So do you think it's structural in that the top layer of the cells is protecting the subsequent layers? Or do you think it's more related to the cellular processes, that there's something profoundly different about these cells and the way that they respond to stress? And if it is structural, have you ever considered a double hit? So, for example, treating with one injury, removing kind of that top layer and then going in again?
Yeah, great question. Have you read my KOA proposal? Is that what's
Actually, no.
No, so I think those...
I should.
Yeah, no, but I mean, I think that those are all great questions that I'm proposing. It's still under review, so you wouldn't have read it. Yeah, I don't know, is it because it's a stratified structure? Is it because there's special desmosomes and intermediate filaments expressed in there, similar to skin? Or is it something else entirely? I think that's another important question we need to understand about the mechanism of this that will have clear, direct clinical implications, as you can tell.
Thank you. I'll stay tuned.
Take one final question.
Nice talk.
Thanks.
And very interesting that in the end you showed an elephant with lots of queries in that. I'm going to add more animals to your kingdom here. You talked briefly about the lungs and the skin. What is your thinking about if you go a little deeper into the skin and you analyze sweat glands? We are in a CF conference here. Sweat gland is one of the good gold standard for the diagnosis of CF. And what your findings are coming to is that it is a stratified cuboidal epithelium. Sweat gland reabsorptive duct is a stratified cuboidal epithelium. It is one of those miniature organs that are very responsive to the injury. Do you think that there are structures like HILOX in the sweat gland, and if it is worth testing and you can learn more from that?
Yeah, very interesting question. And I think it's worth looking into, absolutely. I'll add one thing that's even more interesting on top of that. There's a recent paper out of Elaine Fuchs's lab that looked at retinoic acid sensitivity in the sweat gland and in those progenitors as well. And they have differential expression, different sensitivities as well. So whether there's an actual HILOC with that structure, I don't know. But are there other cells that have some features? Quite possibly, worth looking into. Right? Thank you.
Okay, so our next speaker is Dr. Paul McRae. He's a professor in the Department of Pediatrics at the University of Iowa, and he's going to talk to us about shuttle peptides.
All right, good morning, everyone. It's a pleasure to be here, and I'm grateful to the organizers for this opportunity to present our work. I have this disclosure. So I'm going to give an overview on what amphiphilic peptides are and then show some applications with delivery of adenine base editors and CRISPR nucleases and a little part at the end about other cargoes. So this system was developed by Felden Therapeutics, a biotech in Quebec City that we've been collaborating with along the way here. One important point is this is a co-incubation of the cargo and the peptide in an aqueous solution. They're not linked together. So their idea was, why don't we take from nature endosomal leakage domains, things that maybe viruses have, and combine those with a cell-penetrating peptide to deliver cargoes to cells. So these are the two components. The endosomal leakage domain, which has the features there, it's positively charges, forms a helix in solution, and it's thought to destabilize membranes. This is combined with a linker with a cell-penetrating peptide domain, which also has some positive charges and is thought to have membrane interactive properties. And there's publications on this you can refer to. And we have shown that you can deliver multiple independent cargoes to cells using this mixture. And I'm going to focus on these cargoes. So how is this thought to work? I would say we're not 100% sure how this works, but this is the model that Felden has proposed, and I think there's some reasons to believe they're on the right track. So the ELD is thought to have membrane destabilization properties, and in this example, we're using a cargo of GFP with an NLS on it. So if you put the mixture of the GFP protein with the NLS and the peptide on the cell, within 45 seconds, the cytoplasm turns green. And within 2 minutes, the nucleus turns green. And then how long it will stay, whatever, it depends on the half-life of that protein cargo. So we think there's a very rapid membrane destabilization and rapid entry of the cargo to the cytoplasm. We've done this exact same experiment on ALI cultures of human airway epithelium and basically see the same time course there. So it's a very rapid entry property. And then what happens? Well, as shown in the figure here on the right side, the peptide stays associated with the membrane, and shown in the Western blot, it's degraded over a period of hours. And then in this case, the GFP is in the nucleus. So I want to give a little background on the first experiments we did with this peptide. We similarly took either a GFP cargo or a peptide that was tagged with Cy5-fluorophore and used a prototypical peptide they developed that we're calling S10 here. And you can look at the sequence in the publication. So we applied this to human airway epithelium and ALI culture. The nuclei light up rapidly. And if we put the Cy5 labeled cargo into the airway of mice, you see the cells lighting up throughout the respiratory tract. To look at this a little more in terms of cell types that are targeted, we used the ROSA 26 MTMG mice and delivered a Cas9 RNP that would cut out the red fluorophore and allow the cells to turn from red to green. So we gave 2 administrations of the materials on day 1 and day 2, and at day 7, we looked at the histochemistry. And so what we saw were green cells appearing in the large airway and in the small airways, as shown here in those pictures. And if we quantified that, we saw around 10+% of the cells edited in those 2 regions. Felden also did a duration of expression study in the AI9 mice where they used the MAD7 nuclease delivered with the S10 peptide and then followed cohorts of mice over time for out to 12 months. And there was an average editing efficiency of around 8% that persisted over that 12-month time. And you would expect maybe by a year there's been some turnover of the surface cell types. So one of the first things that we did in our interaction was to see if we could make better peptides for delivery to human airway epithelia. So extensive screening was done with a library of peptides that they generated, and we delivered Cas9 RNPs to multiple donors of human airway epithelia and looked for indels as a readout for how well the peptides were delivering the cargo. And at the end of this, we identified 2 peptides with improved delivery over the S10 peptide. And here's some features of these modifications. And these sequences are also published in a paper that just came out with our collaborators. So S10 is on the top, and if you look at its net positive charge, it's plus 10. The S315 is plus 8. And interestingly, with the S237, the cell-penetrating peptide motif was dispensed with. So this is a shorter peptide, does not have a cell-penetrating peptide component to it, and it has a slightly less cationic charge. And we think this is important for cargoes that involve nucleic acid, because if it's too positive, if there's too much charge interaction, there seems to be a relationship to how much charge there is and how well it interacts with nucleic acid to deliver the cargo, I guess I should say. And by reducing the charge, we think that has a beneficial effect. So let me show you some data with base editor delivery. So here we're delivering the adenine base editor ABE8E as a ribonucleoprotein complex. Our thought there is that that could potentially repair approximately 50% of the CFTR mutations. And we focused on premature termination codon mutation using R553X as an example here. So in the case of R553X, our goal is to edit the adenine shown here back to a guanine and restore the arginine codon. So to do this, we delivered the ABE8E RNP to primary cells that were compound heterozygous with the R553X mutation, and we compared the delivery with S10 and the S237 peptide. So this is a short circuit current tracing that I'm sure you've seen at this meeting. So we deliver forskolin and IBMX to open the CFTR channel, and we apply GlyH to inhibit the channel. We don't see current responses in the untreated cells with 2 stop codons. This is our non-CF control response. With the S10 peptide, we get some significant correction, and with the S237 peptide, we get improved correction, and this is quantified here. When we looked at editing, with S10, we got around 2% editing, and with the 237, this increased to 9% editing on average. Now I wanted to show some in vivo results, and I'm going to present data from 3 models, a non-human primate, a pig model, and a mouse model. So this work with the primate model was done in collaboration with Alice Tarantel's lab. And we did a single aerosol administration of 1 milliliter of the materials shown here. There's 2 experiments. The first one is using the Cy5 labeled peptide. And the second one is to deliver the ABE8E RNP. And in work with Greg Newby and David Liu's lab at the time, we decided to target the CCR5 locus and put an edit there as sort of a safe thing to do in an animal. So with the first experiment, we're going to look at the biodistribution and the cell types targeted, and then in the second one, we'll look at editing by high throughput sequencing. So this shows the Cy5 labeled peptide distribution, and you can see it's targeting cells throughout the respiratory tract, as shown in those white cells on the top panels. And if we look in more detail with some co-labeling, we see delivery to ciliated and non-ciliated surface cells with these markers. If we look for basal cells, it's hard to find a labeled basal cell. We found rare basal cells with signal. And in the alveolar epithelium, we also saw type 2 cells that were targeted. Here, Katerina Kolonkova in the lab came up with this diagram of the airway tree of the monkey here. So on the top left, it's color-coded, and she took samples from all of those regions and then quantified the percentage of cells that had a Cy5 signal, and you can see that in the bar graph on the right. And so we saw a range of 0.5% to 21% of the cells that were positive. And again, this is a single squirt of 1 milliliter into the upper trachea of the animal. So we did not expect to get complete coverage of everything with that kind of delivery. That we think there's a lot of opportunity to improve delivery with these materials. So how about with editing here at the CCR5 locus? Again, using the same color-coded scheme, we did brushings of the airway and then took the brushed cells, isolated DNA, and did high-throughput sequencing, and saw a range of editing from 0.2 to a little over 5% editing. And the lower panels show the controls. There was a little bit of signal with the RNP alone. So here's the pig experiment. We started out with the idea that we didn't have a gain of function pig, so we were trying to do a loss of function by knocking out GFP, so we're targeting GFP. The short answer on that, the GFP expression is so heterogeneous, we could not really discern anything by microscopy. But we looked at the biodistribution similar to the rhesus experiment, and we looked for indels at the GFP locus. So here's an example on the left of the distribution. And I forgot to say, we put this bronchoscopically in the right upper lobe bronchus of the pig. And on the left here, you can see the distribution of the Cy5 labeled cargo delivered with the 237 peptide. And then we've done some co-localization. We see this in secretory and non-secretory cell types at the cell surface, at the epithelial surface. And in quantifying this, we saw improved delivery with the S237 peptide. So improved editing with the S237, I should say. The next model is the GER10 mouse. This is available from Jackson Labs. This has a disrupted venous gene with a stop codon in it. And if you use an adenine base editor, you can restore the venous gene function. So that was our goal. So we gave the peptide and the ABE cargo on 2 consecutive days using the 237 peptide versus the RNP alone. And then about a week later, we looked for recovery of venous expression. So you see on the left the RNP alone versus the RNP with the peptide on the right. And so we were able to see edited cells when we co-delivered the RNP with the peptide. And here's a higher magnification image with some colocalization. Using acetylated tubulin is the red there. And we saw a signal in ciliated and non-ciliated cells. Definitely we're seeing signal in secretory cells. The last thing I wanted to mention was the potential of using the peptides to deliver cargoes, other cargoes, in this case, antisense oligonucleotides. And you've heard at this meeting about applications for these for splicing mutations. For example, the 3849+10 mutation. Another example is exon skipping, such as the work of Hastings et al. And so we use the example of the 1282X mutation with the goal of skipping exon 23 by delivering 2 PMOs identical to those reported by Hastings et al. And through the help of Scott Randell, we got cells that are W1282X homozygous. So we applied the materials for 15 minutes and then removed them, and then looked at restoration of chloride current a few days after that. So here's untreated cells. Here's the cells with the 2 PMOs, and here's the cells that had the S10 peptide co-delivering with the PMO. And if we look at the quantification of that, we saw a significant increase in the signal for chloride current if we delivered the PMO with the peptide. So we wanted to see how this might translate in vivo. So we used an available model called the EGFP654 mouse. This is a splice skipping reporter mouse that you can see here in the diagram of the pre-mRNA. If a PMO targets the 5' end there, you can rejoin the EGFP cassette and you'll get again EGFP gain of function with the skipping. So here in the lower right, you see the cargo alone, the PMO without the peptide. We see very little evidence of exon skipping, but with the PMO and the S10 peptide, we see nice expression of GFP. So I hope I wasn't talking too fast here, but here's my concluding comments. The first is that the shuttle peptides can rapidly and efficiently deliver multiple cargoes to airway epithelia, and I've given you all of these examples. The other thing is that small modifications in the peptide sequences have substantially enhanced delivery of these cargoes, and it appears that the cell-penetrating peptide component is dispensable, which was quite surprising to us. And in general, this peptide-mediated delivery targets the cell types that are accessible from the luminal surface. So the cell types that are targeted are going to depend on the anatomy of the region that your delivery goes to. And the editing efficiency with the ABE8E RNPs were sufficient to restore CFTR function. And I'd like to acknowledge all the help of the folks on this slide, the people in my lab, our collaborators at Felden Therapeutics, other colleagues at the University of Iowa, our collaboration with David Liu and people from his lab, Colin Hemez, Greg Newby, who's now at Hopkins, Alice Tarantel helped with the rhesus monkey experiment, and Steve Murray and Kathy Snow helped us with the GER10 mouse experiment. The University of Missouri Swine Resource Center is our pig collaborator. Scott helped us with the cells I mentioned, and Our friends at IDT have also been very helpful over the years. And I'd be happy to take any questions you have. Thank you.
Hey, Paul, that's
Hi.
really beautiful work. Kate Coffin from Spirevant. I am sort of a fan of these kind of peptide things and cell-permeable peptides that get things in, and it's remarkable that it's not required. But what my question is, you show very beautiful delivery of what you're looking for. Is there a risk that other things, so since it's not conjugated, is there a risk of bringing other things into the cell? And do you have any assays to know if, for example, you just put some other plasmid there with a different marker at the same time, something else that you could see if there's co-delivery because these are not conjugated?
Yeah, that's a good question. You're not the first person to ask that question. And it's true that it's not specific for the cargo. So if you put an ABE RNP out there and you put some GFP protein out there at the same time, those will both go into the cell. And so you could imagine, other cargoes out there or other proteins that are in the lumen may enter the cell. It does beg the question of, how much sampling goes on between the environment and the cell anyway. But it's a good question, and this isn't specific to the cargo. We've done some general safety studies in animals, and don't see any kind of persistent inflammatory response or histopathologic change or cytokine expression that persists or anything. But your point is well taken.
Thank you.
Hey, Paul, that's really great. Lots of stuff there. So with the oligos, I had a question about have you looked at the splicing efficiency of these? I'm always curious about the correlation between sort of the protein or the fluorescence and the actual splicing, either in your 654 model or with the W1282X.
Could you say that again?
Have you looked at the splicing efficiency with the antisense oligonucleotides?
Have we quantified it?
Yes. Yeah.
Not at this point in that last example that I gave. We haven't done that yet. We just went straight to the end point to see what how it affected the net gain in chloride conductance in current.
Yeah, great. I mean, it's impressive to see how much you're getting there, and then it would be really interesting to see how it would work in, for example, like the humanized W1282X mice that they have at Case with Craig and Mitch.
Yeah, there's a lot we can do with this. This is kind of a very preliminary experiment I put in at the end to emphasize that this could work with other kinds of cargoes, but there's much more to do with it, and I appreciate your comments.
Great, thanks.
Hi, Paul. That was really amazing, as always. Two questions for you. One, well, first, I think it's really cool that you've tried so many different cargoes, and so I was wondering if you see any differences in delivery efficiency depending on the cargo, right? Because you have a range of sizes, et cetera. I wonder if you've quantified that.
I can't say that we've done an extensive comparison of that. We do see that certain cargoes are delivered better with certain peptides, and that's the most obvious thing that I could say, we've seen. So we were able to improve the RNP delivery, or a cargo that has nucleic acid associated with it, by modifying the charge density of the peptide. But I would say it's actually rather remarkable that over a huge range of molecular weights, you can efficiently deliver cargoes with the peptide. So, you can deliver antibodies, you can deliver, the RNPs. So there might be very interesting biologic questions you could address using this as a tool to, therapy aside. So
Absolutely. And then, sorry, quick second question. Can these be adapted for systemic delivery?
Say that again?
Can these be adapted for systemic intravenous delivery? Because you've only done airway, right?
I didn't.
Stomach.
Oh. I think this could potentially work at any surface where there's an interface. So I haven't done anything with that, but Felden has shown that it can work in the retina. They have a clinical trial starting in the skin. I don't know if they've done any enteric applications, but yeah, I think there's. But if you were to take this and administer it systemically, you would get nothing because the components just become diluted.
Sure.
So they have to be concentrated at an interface.
Got it. Makes sense. Thank you so much.
Sure.
Thanks very much for that talk. My question is related in that can that peptide be combined with another carrier molecule? Should the properties of the intended cargo not match up with being free?
Yeah, so I haven't done this work directly, but I know that David Gee and colleagues at Felden have covalently linked ASOs with the peptide and shown nice delivery that way. So I think you could improve the specificity with that kind of approach, but with the work we've done to date, that hasn't been the focus.
Thank you. Great talk. I just have a quick question regarding you had said that there was no long-lasting immune response to the RNPs delivered through these. Could you talk a little bit more about if there are like any memory responses if you have done subsequent dosing and seen drop of efficacy due to any immunological memory, or kind of?
Yeah, I think those are good questions. Not in our work, but what I know from our collaborator is they've tried to immunize mice with the peptides to, get an immune response to the peptide and find that, even with adjuvants, it's very difficult to develop an immune response to the peptides. Maybe that's because they're so rapidly degraded. But of course, the cargoes that we're delivering, you might expect that there could be immune responses to those, and that could influence your ability to re-administer them. We have not done work in that area yet.
Thank you.
Terrific. Well, it's working? Yeah, terrific work, Paul. It seems you're perhaps 1 or 2 steps away from human administration. So what's the toxicity associated with these molecules, since there's lots of different applications, but I'm not seeing toxicity profiles. I was wondering.
Yeah, we've published some toxicity studies in a couple of these papers that I mentioned here. As you might expect from the antimicrobial peptide field, these things are cationically charged. So if you put on higher and higher doses, you will see toxicity from the peptide component. But in the range of concentrations that we use for this delivery, we can see a transient increase in cytokines in the lung tissue and a little bit of inflammatory response, but it's all gone within a period of days afterwards, and we don't see untoward responses in the animals. So I think, that's pretty preliminary, simple stuff, but it does look favorable.
Paul, I can't resist. Are you able to get into ionocytes?
Why do you want to know that?
Ian's in the room, that's why.
Yeah, if you think about it, we're doing this on primary culture. So how many ionocytes are in one air-liquid interface culture? A handful. We haven't extensively looked for that. I don't have any reason to think that it wouldn't go into ionocytes, but we don't know yet.
Awesome. Thanks.
Yeah.
Wonderful
Yeah.
talk.
Thank you.
So we're going to move on to our final speaker in the session. Paul is tall. Here we go. Finn Hawkins, who is an associate professor here in Boston. And he's going to talk about transplantation of pluripotent stem cell-derived basal cells. If I can get his talk to start. Thank you. There you go.
Thank you, Amy. Thank you to the organizers for the invitation. It's a pleasure to be here. And I'm going to get started with my presentation. So no disclosures. I'm going to start by saying that I'm representing a much broader team here. So I'd just like to acknowledge the work I'm going to show you is the product of many different groups, but my colleagues at BU, Daryl Cotton and Andrew Barakal, and a very talented group of MD, PhD, and PhD trainees, Martin, Kevin, and Jake, and then our collaborators at Yale, Marie Egan and Emanuela Bruschkia, and their fantastic team. And we've had a long-standing collaboration with Brian Davis, Shingo Suzuki, and Scott Randell. And I'd like to thank the Stem Cell Consortium, and I'm going to get down to business now. A background slide on induced pluripotent stem cells. So induced pluripotent stem cells, or iPSCs, are a really remarkable biological discovery. In 2006, Shinya Yamanaka discovered that by just transiently overexpressing 4 genes in any cell, a blood cell, a tail-tip fibroblast from a mouse, you can convert that cell into a cell that is essentially an embryonic stem cell. And that's just pictured here. Here's mouse tail-tip fibroblasts that were reprogrammed into these iPS cells. We were very excited about embryonic stem cells a couple of decades ago. We all know the ethical and practical limitations of using embryonic tissue, but the reason we were so excited was because, in theory, that small population of cells in the very early developing embryo are remarkably powerful. They're truly pluripotent, meaning they can give rise to every cell type in the body. So if we could leverage that, we could, in theory, make every tissue we want in a dish. But embryonic stem cells are problematic, and that's why iPSCs are so attractive, because we could make them from any patient. And the most compelling experiment for me that illustrates exactly how powerful these cells are is depicted here. So if we were to make iPS cells from a tail-tip fibroblast of, say, a white mouse, and then inject those into an early blastocyst embryo from a black mouse, and then insert that blastocyst embryo now into a pregnant female, we get chimeric mice, where part of all their organs are actually generated from these iPSCs, including their lungs. So if we could actually figure out in a dish how to get those cells from an iPSC to a lung, we would be able to do a lot of remarkable things, from disease modeling to maybe one day even considering the use of these cells for therapeutic purposes. And actually that day is here, because we have clinical trials ongoing with these cells being used for therapies that you would never consider possible, including inserting these cells in the brainstem of patients with Parkinson's. So they're pluripotent stem cells. There is another remarkable type of stem cell, and in the lung, the adult stem cell in our airways is called the basal cell, as you've heard about today. This is pictured here in this pseudostratified airway epithelium of the human airway. You can see these basal cells line the basement membrane. And these are remarkably potent cells. So as many of you have had flu or COVID in the past couple of years, when your airway is injured, these cells can remarkably get activated, proliferate, and differentiate into all the specialized cell types. And they are really potent. I was really struck by Virel's comment that, one hillock basal cell clone can give rise to 10,000 progeny. I mean, that's really remarkable. So basal cells are stem cells of the human airways. And so the question is, back to our chimeric mouse, can we actually figure out how to get cells from this pluripotent stage into a basal cell stage? And I'm going to summarize all that work. It was about 14 years of work that we undertook, myself, Daryl Cotton, Shingo, and Brian, and I'm going to save you the details of the protocol, and instead I'm going to try to prove to you that we did actually make those cells. So one of the nicest, cleanest assays that we do is stem cell biology. is we do a clonal assay of function, and we simply ask one single cell, what can you do? And that's what we're doing here. We're taking one single iPSC-derived basal cell, and we're following it in time as it grows in a dish, and it forms a multicellular organoid. And it tells us that it can actually undergo self-renewal. So it's given rise to many more basal cells, as you can see here in green. But it's also undergone multi-lineage differentiation into specialized cell types. So it's given rise to secretory cells and ciliated cells, and it's therefore fulfilled the criteria for an airway stem cell. Some remarkable properties of these iPSC-derived basal cells, or IBCs, we can propagate them in culture for years. We can differentiate them in air-liquid interface culture like we do for primary cells. They form these beautiful mucociliary epithelia. And they can be efficiently cryopreserved and then used again. And just remember, these cells came from a single blood cell from a human individual 10 years earlier, and here they are repurposed 10 years later to make airway epithelium. So could we actually one day apply the potency of the basal stem cell as a cell-based therapy? And I think the overall concept that I've been thinking about a lot is that the basal cell actually represents a vector in and of itself. It's a cellular vector that's intrinsically programmed to be able to propagate throughout the airway, differentiate into all the specialized cell types. And in the case of CFTR, it has the potential to propagate that normal sequence throughout the full airway epithelium in the right cell types. And not only that, but then have the regulatory equipment to express it at the right physiologic levels in those cells. And the overall two approaches we think about are that we could do it from primary cells, so autologous, that if I had cystic fibrosis, some of my cells could be isolated, expanded, gene-corrected, and then delivered back into my airways, or through the iPSC approach, as I just mentioned. But this is obviously looking very much forward into the future as a potential durable cure for CF. So we started with some much more feasible, simple questions, the first of which I'll talk you through now. So can we do this in the simplest form in a mouse? And the experiment here is first we're delivering a detergent polydocanol into the trachea of mice, which is a good model of the small airways of humans. The polydocanol will cause an injury to the epithelium, as you see here. By 5 hours, we just have a few basal cells hanging on for dear life. But those basal cells are very powerful, and just 2 hours later, they've already spread out over the epithelium. And so we're picking this 5-hour time point to now deliver primary basal cells labeled with GFP so we can easily track them, delivered into the mouse at the 5-hour time point. And similar to Biral's data, we're now going to excise the trachea after at least 4 weeks. We're going to fillet it open along its longitudinal axis, and we're going to first ask, are there cells? And without any injury, not surprising, the trachea is very effectively equipped to get rid of anything that you put in it that's not supposed to be there. So without injury, there are no cells. But in the context of injury and cells, you can actually get very robust engraftment. And so this, on average, is about 30% engraftment. And then if you perform single-cell RNA sequencing to ask, well, what are these cells? If we look at the whole tracheal epithelium, you can see all the expected cell types: basal cells, secretory cells, ciliated cells, and then the more specialized cells, neuroendocrine cells, and ionocytes. And the question is, are our donor cells actually contributing to all those lineages? And the answer is yes, they are. And even more surprisingly, there are no molecular differences between the donor cells and the endogenous cells, despite those cells having spent weeks and months outside of a mouse in our lab. They're indistinguishable. So what about stem cell-derived basal cells? So I showed you our human data earlier. We've been able to adapt that now to mouse ES cells and iPS cells. And so in this experiment, we're now transplanting mouse embryonic stem cell-derived basal cells and asking, can those engraft in the exact same injury model, also labeled with GFP? And actually they can, very similarly engraft. They give rise to all the specialized cell types. They also replenish the basal cell stem cell pool. That's essential as we think about stem cell therapies, for the lung, the ability for those delivered cells to be able to durably persist, respond to subsequent injuries, and maintain that epithelium over time. And we also performed single-cell RNA sequencing to understand what these cells are doing over time. And so you can see here endogenous versus the donor cells at 3 different time points. And I'll just highlight, it's a complicated UMAP, but really here in the center in the pink and green is where our donor cells are. And there's a couple of really notable things to highlight here. First is the cells are intrinsically programmed to generate the distribution of cell types at about the appropriate frequencies, which is quite interesting. The second thing is our iPS or our ESC-derived basal cells have given rise to all the expected cell types again, and molecularly they express all the genes you would expect them to express. And then the final surprising thing is, despite all that, they are still molecularly different to the primary basal cells, even after 192 days in the mouse. And those differences represent, we think, some regional differences in terms of proximal distal airways and some differences in immune signaling. And this is one of my favorite images. This is a trachea of a mouse 2 years after transplant. And you can see there's robust engraftment. Viral, I wonder if these are some of your Hillock cells that have expanded after the injury. But this mouse was fine. At least he didn't submit any complaints to us. And the reason I like this is because I like to dream big about CF patients and to think that these cells persisted for the lifetime of a mouse. If we could do this in humans, how long could those cells persist? Another assay that we really like in the stem cell field is, in terms of understanding a cell's true potential at regenerating an organ, is to do serial transplantation. So we ask, can you regenerate an organ? Okay, we'll take you out again. Can you do it again? Can you do it again? And so we did that with our ESC-derived basal cells, and you can see after the first transplant, isolated, expanded, retransplanted, they were able to do it again, and a third time, and a fourth time, and a fifth time, and a sixth time. And I believe Daryl Cotton has told me that it's currently in an 8th generation mouse, which is very surprising to us. We'd love to understand more of this biology. This beats the hematopoietic field. We'd love to understand why. You can also engraft human basal cells. Here are primary human basal cells now in an NSG mice, because obviously we need to suppress rejection, but they also engraft a little less efficiently. They differentiate. They're easy to spot in the mouse because they're larger and they form goblet cells. And our iPSC-derived basal cells can do the same. This was the initial experiments we did that are in our paper from last year, which were a little less efficient. But the cells engraft, they give rise to all the expected cell types, including goblet cells. And for me, I've spent a long time developing these cells, wondering, am I really making lung? Just looking at some markers, and then you see them in a mouse functioning, and you think, okay, they got there eventually. And one of the things we're really excited about this approach overall is that now you could actually, as we think a lot, we've heard so many talks about delivery, the challenges, the models, the animal models, the limitations, the benefits of each. But one of the potential applications of this technique now is that we can transplant human cells, for example, from a CF patient, or in this case, a PCD patient, into a mouse trachea and form like a humanized chimeric trachea that you could use to now study delivery. But I've shown you a lot of data about differentiation and persistence, and now it's time to ask whether we can actually restore function and cure diseases. And so I'm going to share with you some preliminary data led by Andrew and Kevin here, and in collaboration with our Yale colleagues. And the question here is whether you can reconstitute CFTR function in an animal model. And we're starting small here in the mouse, but we have big dreams. So we've taken the CF gut-corrected knockout mouse, and then we're taking primary basal cells, and labeled with GFP with wild-type CFTR. We're transplanting them in, exactly as I showed you before. And we're going to ask three questions. We're going to ask, are we getting CFTR message? We're going to ask, are we actually getting restoration of CFTR current by performing an Ussing chamber analysis of the upper trachea? And we're going to ask, if so, what are the cell types that are contributing to that CFTR function? So here's one trachea. I'm showing you one of the best tracheas, but we had several very good tracheas. You can pretty much make out individual cells here, which is, I think, is just such a beautiful image. And I just love this image because this, to me, looks like a therapeutic, right? I think of this 10 years, 15 years from now, that these represent a cellular therapeutic in the airways of our patients. And so let's look for message, and I'm just going to highlight in this, this crisp gel here the key samples to look at. If you look at our CFTR knockout mouse, you can see that there's no GFP because there was no transplant, and there's no CFTR. But here in this purple box are the CF mice that did get transplanted with the wild-type cells, and you can see that they all have evidence of donor transplants with the GFP, and 7 out of the 8 had evidence of CFTR mRNA. But we wanted to see function, and we were hoping for a little blip, some signal that these cells could actually restore CFTR function. I'll show you one representative Using chamber here of the upper trachea, where you can see compared to wild type, or the CF control, we actually have quite surprisingly good restoration of CFTR-dependent current. And then when you look at that over the mice we've done so far, you can see that actually there's no significant difference between those mice and the heterozygous controls. So again, this data is fairly fresh off the press, but we did single-cell RNA sequencing, and we're just going through the results. But what's become clear to us so far is we have clearly reconstituted the different cell types, shown here in this UMAP. We have a rare cluster of cells here, including ionocytes. And if you look at GFP overlaid on the whole UMAP to understand where the donor cells are, you can see that some of the donor cells are in this ionocyte cluster. And if we look to see if those are truly ionocytes by looking at a panel of classic ionocyte markers, we can see that they look very, very much like fully functional ionocytes, and they're the main cells of our transplanted cells that are expressing CFTR. All right, so what I've shown you so far we think is extremely exciting. We've spent years and years building up to this point, but we also recognize that it's proof of concept, it's a first step, and really it's a very small baby step when we think about the challenges of doing this for a human lung. But it's that first go signal for us that this might actually be feasible. Where do we go next? There are many, many hurdles, but a very big hurdle that we want to now start to tackle is preconditioning. How do we move from our very brute force polydocanol approach in a mouse to start thinking in a much more sophisticated, translational, targeted, precise way to injure the airways and deliver these cells? Anat Cohn here is thinking about ways to target the delivery, starting with a mouse model. But the path to a cure includes some really important milestones for us, including we need to move into a larger animal model to ask about feasibility and ultimately whether you can rescue disease phenotype in a larger animal model like a ferret. We think a lot about preconditioning, and we think this is where we'd like to spend the next several years of our careers working together to try to move this forward. So I will stop there and take any questions. I acknowledge the team, but we've been here.
Super exciting work. We have plenty of time for questions.
That's really exciting. Hi. So I have just a quick, probably kind of ignorant question, but are the resident cells, the mutant cells, do you anticipate that they would cause a problem by, you put in the new cells, but you're going to have still the resident cells, right, that are going to be having potentially mucus, right?
Let me just see if I fully understand the question. Can you just
I'm just saying if you transplant in the cells, right, and it looks fantastic, right? You have a lot of cells in there that are functional, right? But what about the cells there, the resident, the native cells there that are still don't have CFTR, right? So they would be still producing mucus, possibly, right? Do you see that as being, or any other thing from the resident cells that might cause a problem?
Yeah, we don't know the answer yet. I think it's the same question we have for all the other vector therapies as well, as how many cells do you actually have to hit? How many cells have to be corrected to overcome the remaining cells that still remain diseased? I think it's a key question that we need to figure out for all the various gene and cell-based gene therapy approaches.
But I guess in your case, you're not correcting any of the cells that are there, you're just adding cells.
Yeah, so the idea would obviously be for CF, would be to correct the cells ex vivo.
Right.
Yeah, so you'd be putting in corrected cells. they'd be autologous.
No, yeah, no, I
gene-corrected
understand that.
cells.
But you're not correcting any native cells necessarily. Got it. Yeah.
Not the endogenous cells, no.
Right, endogenous.
Yeah.
It was a lovely talk, thank you. Excuse the naive question. Could this be delivered systemically, either as iPSCs corrected or basal cells corrected, rather than down the airway?
Yes, so we're taking the very first steps towards what I think is an entirely new discipline of trying to figure out ways to actually precondition the lung. So I don't think we know yet. My personal favored approach, and this is just speculation, is that I like the approach of targeted, localized, controlled endobronchial delivery. However, there are others who have pursued intravenous approaches, and this field is very nascent, but Yair Reisner is taking an intravenous approach to deliver cells. Our favorite approach would be more targeted delivery to the airway epithelium. We think that's probably going to be the safer approach.
Go in center.
Very nice talk.
Thank you.
Do you think that the wild-type cells that you are implanting in the CF mouse could eventually, with time, replace the native cells because they have selective advantage because they are physiologically more apt for the airways, or do you think that won't happen?
That is such an important question, and I don't have the answer yet, but I hope in a couple of years I will have. If the signal is that the corrected cells have an advantage, that's very exciting to us, but we don't have the answer yet. We think about a lot of ways to impart a competitive advantage, at least initially, but we don't know yet.
Thank you.
Yeah, absolutely beautiful work. It's a spectacular approach to such a disease and change. And I'm just wondering, right now you're saying basal cells for iPSC cells. Is there a vision to use a different cell source, or do you think for people who would potentially undergo something like this, would you have to get cells from the lung, or would you start for something else? I'm just wondering what the starting would be.
Yeah, I think so. First of all, I think gene therapies are going to come first. But in terms of cell-based therapies, I think autologous primary cells are going to get through the FDA faster. So that's likely going to be autologous primary basal cells. I don't know how long it will take for the iPSCs to get to that point, but what we're excited in terms of iPSC-derived basal cells is the serial transplantation might suggest that they're rejuvenated. They are really potent basal cells. And then I guess the Hillock cells have been fairly recently described, and I think we don't fully understand yet all the basal cell populations in the lung, but I do think the basal cell population is the most compelling cell to target.
So you would get that through a bronchoscopy or something?
Yeah, I think that would be the idea, that you would harvest basal cells via bronchoscopy, repeated brushes, and then you would expand those cells ex vivo. And this is thinking, quite far down the line, but these are the type of experiments we're trying to think about for larger animal models. Yeah.
It's beautiful.
Incredible talk. Thank you so much. So you had mentioned that there were minor differences in expression of your basal stem cells compared to the normal endogenous stem cells. So in your DREAM experiment, we have 30% now preconditioned, selective advantage, and now every cell is now edited and creating CFTR. Do you think those small differences will eventually become something larger when we think of location, tracheal versus distal airspace, or anything like that?
Wow, that's a really tough question. It's a very good question, but I don't know. I think we just have to do the experiment.
You're speculative, it's, do we start to see a growth of these small changes become something bigger if that does indeed take over the entirety of the lung, or that's our predominant producing cell at that point?
Yeah, I mean, it's possible. I guess I'm pretty reassured by our 2-year data that at least functionally, if there are molecular differences, the mice seem to be doing just fine. Where it will get more complicated is the larger animals, where there's a lot more complexity of the airway epithelium from proximal to distal, and it's relatively simple in the mouse. Those, I think, will get more interesting.
And the strange tweaks in humans that we always see, that there's always that slight difference.
Yeah, exactly.
Okay, great. Thank you.
So a quick follow-up question. Have you seen evidence of your transplanted cells going into the alveoli and becoming alveoli, or being rogue basal cells in that space?
Yeah, that's a very good question, and we have. We see that if we deliver our cells and there's been injury more distal in the lung, the delivered cells can attach, but they do not differentiate into alveolar type 2 cells that we've seen at all. They seem to retain their airway program. But what that tells me is that as we seek to develop these targeted preconditioning regimens, they need to be very targeted. The alveolar epithelium needs to be left alone so that the donor cells go to the right place.
And then I have one other quick question. You're talking about the competition potentially in vivo. Have you done any competition assays with your ibasals and other cells in differentiated airways in vitro yet?
We have not. I think others have, and I don't think it's published yet, but we have not done that yet. No.
Great.
Super exciting work. Thank you so much. Thanks everyone for coming to the session.